Okay, good morning. Thank you very much. What I thought I'd do is talk a little bit about an experiment that we put forth in California called the State Stem Cell Agency, which was really an experiment where actually the citizens of California put on the ballot a proposition to study a new technology, stem cell research. And it was a $3 billion bond initiative that was created in 2004 when the federal government policies did not allow study of a particular type of technology, human embryonic stem cell research. And so this was a state entrepreneurial effort where what we're doing is collaboratively developing scientific, regulatory, and commercial pathways, and also funding the research to produce stem cell-based therapeutics with the focus to do the training, but also have a very concentrated effort to see what we can do to accelerate as much as we can the introduction of a new technology to develop a new type of therapeutic. This required partnerships across a broad swath of the community, including industry, academic support, and patient advocacy partnership. The $3 billion was really used as a stimulus to the academic and the biotech sector to build research excellence, both within the emerging biotech industry in stem cell biology, as well as in the academic community. And here the real focus is to support the best science, the best scientists, and the translation, as well as the discovery. The driver for this, like any new technology, is the promise of improved patient outcomes, the promise of quality of life improvement, the promise of reduced health outcomes, reduced healthcare costs, and the promise of commercialization of a new technology. So all of those were drivers. So basically, because of time limitations, I'm going to focus more on the big picture. But the first 5 years of the Institute was really focused on the discovery engine and making sure that we seeded the field to make this technology, to have the seed grants, to train the people, to bring in the research leaders. The current 5 years is really one of prioritization and focus. And with an aim in mind, I mean, this very cognizant of what was just discussed by Bill about science versus engineering. We're trying to merge the two to some extent, and maybe that will be a provocative discussion later on about how to do that. These are just some of the activities. We're working actually with 21 different countries, states, foundations, and the NIH. We have hundreds of collaborations. 70 of our programs are actually in the translational space. The vast majority are still in the basic and early discovery space. But in those programs that are moving forward, we're actually working very hard, not just on the funding, but on thinking about what the pathway is to move translational research forward into potential therapies. This involves partnerships with academia, really looking to academic infrastructure for the innovation and the solutions. So it's looking not just obviously at the infrastructure, but looking at the research capacity and the innovation. But it's also looking at industry, not just for their big pockets, but because of their manufacturing, because of their standards, because they do have a model, perhaps more based on engineering, about milestones, go, no go, success criteria, and the relevance of the type of projects that we're working on, and does that have a chance of being competitive in the market landscape. And then again, thinking of the future with doing clinical trials and partnering with patients, with the clinical infrastructure, making sure that that is really orchestrated and organized to actually be able to evaluate an innovative technology. There's some very unique aspects to the type of therapy that we're working with. These are live cells. Lots of problems with putting these into preclinical animal model systems. They're all xenografts. So we have to do quite significant manipulations of immunocompetent animals or use incompetent animals. Or what we're also doing is actually developing human-induced pluripotent stem cells as tools for actually understanding the disease mechanisms and looking at it for drug discovery and development. We work around the world. These are just some of our collaborations that we've set in place. We're putting in place the tools to actually think about what are some of the unique translational hurdles to overcome for stem cell therapy. With this, we're looking at replacing diseased or damaged tissue. These cells, many of them have to live, they have to survive, they have to engraft, and they have to functionally integrate. And how do you measure all those things? We also need more predictive preclinical modeling. Right now, with With the animal studies, as I said, they're all xenografts. We have problems with xenograft retention, working on those ways to induce immune tolerance so that these cells can be accepted. And we have to think about these things because we have to show to regulatory agencies that we've got enough data and we've got evidence to move these into patients. In addition, we have to look at ways to actually track these cells in terms of imaging them or different types of assays to be sure that they're actually maintaining the phenotype that we want them to maintain, that they're not turning into a tumor, they're not differentiating into some other kind of cell. And the other thing, if this is actually ever going to be a therapy, is looking at the cost of goods, at reproducibility of the manufacturing, and trying to develop the tools and the small molecule substitutes for differentiation factors. In addition, with this kind of innovative technology, thinking about the delivery devices for these cells. So this involves not just the therapeutic cell itself, but the delivery device. And so these are all combination products. We have early translation programs. This gets to the issue about, is the data rigorous enough to actually be acceptable to an industry sponsor to be able to take it forward? We actually don't have the time, money, energy to replicate experiments over and over again. So what we're trying to do is induce a level of rigor when we do these experiments, at least when they're at this stage, so that hopefully it will reach a standard that's usable by those other than those in the laboratory. We also are looking at moving those types of programs into the clinic and considering the types of issues that need to be thought about as you're moving a product into the clinic, and really making sure we're answering the right questions, that we're doing it with a level of rigor that it actually will be usable by an industry partner, and looking to industry not just for their big pockets, but for their expertise in regulatory, in technical, and commercialization issues, so that we are partnering them very early as we move forward. 85% of our projects are with academics, with innovative technology. But a lot of what we do is actually working with the investigator on an area of research that they usually don't focus on. It's usually about scientific discoveries, publishing, getting grants. It's not about developing a product. And so we're finding, at least in California, we're actually working quite extensively to develop a cadre of expertise in also therapeutic product development from the academic center. This is just looking at the slices of the pie. We look at many other things in addition to cancer, but it makes up about, at least in the translational portfolio, about 112 million of the 600 million that we've spent. So those are the slices of the pie in black and in yellow, and they're both in solid tumors as well as in hematologic malignancies. So in addition to funding, a lot of things I think to look about is maybe the training and the education. We're working quite extensively with the investigators to help position them to be successful and to answer the right questions. So we're working very much with them on the science and the evidence, but also on helping to carve a regulatory pathway for a new innovative technology. We're working with the FDA and other regulatory agencies on educations, on webinars, on roundtables and conferences. So it's not just funding the research, but it's also trying to help provide the education that's necessary for the regulatory pathway. And for the regulators, they also appreciate having a neutral nonprofit third party actually bringing the technology to the table so they can have a better understanding of the depth and the issues involved in bringing new scientific technology forward. And it's not just the US FDA, we're working globally with other regulatory agencies around the world. We held a meeting actually last month in Washington, D.C., where we had regulators from Canada, the U.S., U.K., the European continent, and Japan talk about where things are converging, where things are quite different, with donor eligibility, with cell source, with CMC, with preclinical animal models, with risk-benefit issues for bringing an innovative technology into human clinical trials. One of the areas that we're working on extensively and trying to provide it as a research resource is that of the HPS initiative, and that's the Human-Induced Pluripotent Stem Cell Initiative, where we take cells from either the skin or from the blood and actually revert them back to an embryonic state and then mature them to the particular cell of interest. We're taking these from patients with a variety of diseases as well as from controls, and involves a complex series of interactions in terms of donor eligibility, informed consent, what data can be shared and what cannot. We're working with a tissue deriver to actually derive all the cell lines using a standard model of derivation. And then we have a repository, it's Coriell, who's going to actually bank and distribute all the cells. So we're trying to put together, we are putting together an initiative of a research capacity to actually provide the tissue, think about how to derive them, and have a distribution system and how to share the information and data so that we can enhance what we know about disease modeling and target discovery, and also have a research resource with human cells for drug discovery and development, which we think will be a more better mimic of the human condition than the preclinical animal models. These are some of the main activities of the resource with collecting, deriving, storing, and distributing, and they will have implications on one of the policy recommendations I'm going to bring up later. The other capacity building research resource, which we'll also get through during the day, is how to build the right kind of network for innovative technologies. We just put out a solicitation on Friday to build a stem cell clinical infrastructure network that tries to learn from lessons that have already been known with other clinical trial networks that are out there, and try to think of how to design this in a different way so that it can be more streamlined, more effective, and more efficient. How we can think about ways of sharing data and information, not just to those within the assembly of people who are part of the project, but more for the public and the patient. We need to do a much better job as scientists and clinicians in informing the public. And so we think this is a very important thing we need to integrate into our clinical trial networks, and also take into account the healthcare economic issues as we're trying to move forward and build the evidence database to help inform those business strategies and reimbursement strategies. This is just showing you the different circles of the different people that need to be involved in the discussion with academic, regulators, patients, healthcare professionals, and industry. And this is touching upon the three policy points that I'll end with. One is really to enable expanded access to biomaterials and the associated data while ensuring protections and accountability. This is just showing the different partners in that information and biomaterials exchange, from the donors, the patients, the investigators, the commercial entities, and the information and material generators. The second is to really optimize mechanisms for clinical trial review, oversight, monitoring, and information sharing. I have a series of questions here that one could consider in trying to put together such a network and think about the policies that might need to be addressed or changed to put this into place. And then also the issue of innovation. How do we fund and have regulatory approaches that encourage and foster innovation rather than being risk averse? I think this is a really big issue. Because if you want to think about getting something into the system, science has to float, no doubt about it. But can we do something so it takes less than 30 years to get innovative technology into patients? And what can we be doing differently to try and accelerate that? So these are just some of the questions I pose that we might want to consider. So this is just a summary of the three policy priorities, and thank you very much.
We have one or two questions for Ellen. So Ellen, could you talk just a little bit about what you imagine in your first problem? I mean, how would we expand access? What are the particular challenges that you'd like to see policy change?
Well, I think it's about informed consent. It's about donor eligibility. It's about maybe the intentions, maybe repurposing specimens. And when you have informed consent that was arrived at one point in time, how do we think about the issues in terms of being able to reutilize those specimens? I think the other issue is that of privacy protections, the more and more, particularly with genomics data, particularly with certain types of data, how can you prevent individual identification? And I think just maybe it's more public anxiety than real substantive issues, but there's a real issue about losing confidentiality and having data published and out there that's your own. So I think informed consent and how to reuse specimens might be one item to think about.
Great, thank you. It's interesting. Last night we were, the National Cancer Policy Forum met, and it's interesting how many of the topics that are brought forward this morning were exactly some of the things that we were chewing on last night. So I think it's a very, very good discussions. So we're going to move on so that we have time for our panel discussion. I'm next pleased to invite Denise Galloway, from my colleague at the Hutch, to come and talk to us. Denise, of course, has been instrumental in understanding HPV infections and the development of vaccines that have allowed us to really change the course of history within that disease. And she'll be talking about infectious agents, cancer development, and global health. Denise?